My name is Kathy Breheny. I'm here today to do an experiment called isolating cheek DNA. I apologize in advance if there's an echo. I'm in a basically empty apartment down in Florida. I have nobody down here to help me, nobody at all. So I'll be videotaping this and splicing it together the best that I can. So let's get started. The materials we're going to be using for our experiment today include distilled water, rubbing alcohol, which I just took out of the freezer, so it's very, very cold. We have some dishwashing liquid, some meat tenderizer, salt, some measuring spoons, a 15 milliliter tube. We have some pipettes. We also have 2 beakers. One is labeled lysis buffer and the other is labeled Protease solution. And we will get on our way and see what we can make happen. So the first step in our experiment is to make a lysis buffer. So what I've done is I've used a pipette and I've added 1.5 milliliters of dishwashing detergent into a beaker with 8.5 milliliters of distilled water, and to that I added 1/4 teaspoon of salt. Then I stirred it gently until the solution was evenly mixed and all the salt was dissolved. Next, we're going to make our Protease solution. Using your pipette, add 10 milliliters of distilled water to your beaker. Then add 1/8 of a teaspoon of meat tenderizer to your distilled water. Stir the mixture until the meat tenderizer has been dissolved. Okay, so now we have our lysis buffer and we have our Protease solution. All we need are some cheek cells. The way we're going to gather the cheek cells is I've set aside 3 milliliters of tap water in a cup, and I'm going to gently chew on the inside of my mouth for 30 seconds. After 30 seconds, I'm going to vigorously rinse with the 3 milliliters of tap water that I had set aside. After the 30 seconds of rinsing, I'm going to transfer that solution into a 15 milliliter tube, and then we can begin our experiment. Okay, so once we took the cheek solution and added 2 milliliters of lysis buffer to it, and after inverting it 5 times, you can start to see that there's a reaction happening, and you can kind of see it here. There's a suspension of particles in the liquid. And what's actually happening right now is the detergent in the lysis buffer is hydrophobic, so it's breaking down the cellular membrane of our cells. And because those cells are phospholipid bilayers, this is what's happening. So right now, this is how we're getting to the center of our experiment, which is to extract DNA. First, we have to break apart the cell, and that's what's happening right here in this step with the lysis buffer. Okay, so to my test tube, I added 100 microliters of our Protease solution, and I inverted the test tube 5 times. And now I'm just going to let it sit here for 10 minutes undisturbed at room temperature so that the Protease can start doing its job. And we'll come back in 10 minutes. Okay, so what I've done is after letting our test tube sit with the Protease solution in it for 10 minutes, I then took my isopropyl alcohol out of the freezer and I added the alcohol to my mixture at a 45-degree angle. I'm not going to disturb it now, but and basically what I'm observing is that there are 2 distinct layers. Right now, the alcohol appears to be floating on top of the cheek solution with the Protease and the lysis buffer in it. So I'm going to let this sit for 5 minutes, and then we're going to come back and observe the layer where the alcohol meets our solution and see if there are any changes. Okay, so here's what happened. When I added the cold alcohol earlier, there were were two distinct layers between our cheek cell solution, and the alcohol. They formed two distinct layers. Soon after the alcohol was added, I observed strands being pulled up out of the solution into the alcohol. They're gone now after the 5 minutes. They're no longer suspended in the alcohol. It appears that they have, and unfortunately you can't see it here, it appears that they settled into a layer on top of the cheek solution and between the alcohol. So right now I'm going to invert my test tube 5 times, and then we should be able to see DNA strands floating in the solution as it gets mixed. Oh, look at that. You can see that. Check it out. That's my DNA. And that's what I saw earlier. So let me see. Oh yeah, you can see little strands of DNA floating up. It likes to float up to the top. Look at that. There it goes. Wild, and it's right up at the top now. So there you have it. Kathy's DNA floating in the test tube. Pretty radical. So today we set out to extrapolate DNA from cheek cells, and our experiment proved that we could do that. So let's just recap how we went about getting the results that we got. Number one, we started with a lyse buffer, and the lysis buffer was dishwashing liquid, salt, and distilled water. And basically what lysing is the breaking down of the cell. And the dishwashing liquid pretty much dissolved any of the oil-based molecules in the cell. So basically broke down the phospholipid bilayers around the cell. So that was the first part of our experiment, was to start that breakdown of the cell. Second, we created a protease solution, and protease is an enzyme, and that's why we used a meat tenderizer, because meat tenderizer is a protease enzyme. The one that I used is derived from pineapples. Bromelain, I believe the name of it is. So basically, the protease enzyme starts to break down all of the other cellular proteins within our solution, in our cheek cells. The other important thing that happened during this experiment was, because of the salt in the solution, the DNA became insoluble. And the reason that happened was because the DNA has a negative charge to it, usually, and the salt in the solution has the positive charge. So when the two came together, the DNA became neutral, so it had no charge. And when that happened, it became insoluble to the water. So everything else continues to break down and dissolve in the water and is soluble, but the DNA was strong and stayed together. So we then added, finally, some very, very cold alcohol. And the isopropyl alcohol, what that did was it caused the DNA to aggregate or clump together. And that was apparent when we saw the strands of DNA clumping together and growing upwards. And then eventually they all settled down into that little layer between the alcohol and the cheek solution. So basically that's what happened today, and it was a very successful experiment, and I hope everybody had a good time watching. So thank you.